Welcome back to all of you. Maya and Ronen, you talked about the human microbiome and discussed the medical implication of the interplay between humans and the bacteria they host. Following your discussion, you asked me about the next generation sequencing technology known as NGS, which enables exploring the human microbiome. Actually, the application of NGS technology towards the microbiome is somewhat different from the application of this technology to sequencing the human genome itself. So I propose the following. In the first segment, we will talk about high-throughput sequencing technologies, describing the experimental processes involved. Variants of the same technology are used for sequencing the human genome, either at the level of DNA or RNA, and for microbiome analysis. In the second segment, we will talk about the computational analysis, mainly describing how we go from the automated output of the sequencing machines, called reads, to detecting genomic variation in humans. In the final segment, we will talk about clinical application of this technology and return to microbiome sequencing. The technology for sequencing DNA is quite old. In 1977, Frederick Sanger and colleagues developed what we call now Sanger sequencing. Sanger, by the way, won 2 Nobel Prizes in chemistry, the second for this contribution. Using the method he developed, the nucleotide sequence of a single DNA segment could be read. However, the transformation to next-generation sequencing allowed millions of reads to be determined in parallel. This technology has many applications. For now, we will concentrate on sequencing individual human genomes. Since the reference human genome is already known, we can use it as a scaffold and we can look for individual variations relative to the reference. In today's lecture, I will not be able to accurately describe all the methods used, nor their technical details. Rather, in the little time that we have, I will try to convey the general ideas behind these methods. I will start with a slightly older high-throughput technology, although strictly speaking, it is not considered a form of sequencing. We call it microarray technology or genotyping. In one version of this method, we synthesize between 500,000 to 1 million short single-stranded segments of several dozen nucleotides in length, known as probes, with sequences based on the reference human genome. These probes are adhered to a chip. Then we take the DNA we wish to analyze, fragment it to billions of short single-stranded segments, which are then fluorescently labeled and allowed to hybridize to the probes. If a sample fragment is fully complementary to a probe, it will bind strongly. And if it is partially complementary, it will bind weakly. If our goal is to detect large insertion or deletion in the patient genome, as required, for example, for prenatal genetic counseling, we will coat the chip with an array of DNA segments representing an even spacing across the genome. For example, a short probe for every 1,000 nucleotides of genomic DNA. While the lack of hybridization of few probes can be due to technical issues, if we detect a longer series of probes that originated from a continuous region of the genome that have not been hybridized at all, this indicates that the non-hybridizing region is missing in the genome of the patient. In fact, based on the intensity of the fluorescence, we can know whether the deletion is homo or heterozygous. These techniques can also be used to detect specific sequence variation. If the probes are selected to cover SNPs, the positions we described in the previous unit at which there are known heterogeneities in the human population, we can use another version of this technology to detect nucleotides variation of a patient at each tested position. This is the technology still used in GWAS, genome-wide association studies, which we will describe in a later unit. This technology is also the basis for many of the commercial genetic testing kits offered directly to the public by companies like 23andMe and MyHeritage. The next technological advance was the introduction of next-generation sequencing. In one version of the technology, used in the very popular Illumina machines, we start by fragmenting many copies of each chromosome into fragments of several hundred nucleotides each. Then, short nucleotides adaptor molecules are added to each fragment, one on each side. The fragments are allowed to hybridize to the surface of a flow cell that is plated with sequences that are complementary to the adaptor molecules. Then, a polymerase reaction is introduced to replicate or amplify the sequences to 2 copies, then to 4, and onwards until a cluster containing a large number of clones of each original sequence is formed around the original DNA molecule. All the sequences are denatured to a single-strand form, and then fluorescently labeled nucleotides are added to synthesize the complementary strand. Since these nucleotides are labeled, it is possible to trace which one of the 4 nucleotides is being added, and thus to determine the sequence of the original DNA fragment. This process starts from one termini, or in what is called per-end sequencing, to start from both termini. The original methodology enabled the accurate sequencing of a stretch of about 35 nucleotides starting from the termini, after which the fidelity of the process was lost. Currently, we can sequence about 200 to 250 nucleotides. This process occurs in parallel on the chip for millions of sequences, and thus we obtain what we call reads that we need to align computationally to the reference human genome. We can either sequence the entire genome in what is known as whole genome sequencing, or to reduce costs, we can decide to sequence only the coding region of the genome. This can be done by using specific primers to amplify only exons to get what is known as exome sequencing. The number of reads obtained in the experiment determines the coverage or the depth of the sequencing. Coverage is equal to the number of reads times the length in nucleotides of each read divided by the size of the genome or the exome. A high coverage reduces the probability of experimental sequence errors. A coverage of 30 times is considered very reliable, and for this coverage, whole genome sequencing of the human genome with read lengths of 250 nucleotides would require approximately 360 million reads. These reads are stored in a FASTQ files, a format that retains the actual sequence of the reads as well as information regarding the technical details of the experiment, including information about the quality and thus the reliability of each nucleotide identification. You will hear more about FASTQ file format in the practice session. We can also perform what is known as RNA sequencing or RNA-seq in order to measure the level of expression of each gene in a given tissue. This can be done with some variations of the basic protocol. First, we need to capture the RNA content of the tissue. For technical reasons, the RNA is converted into a form of DNA known as cDNA and then sequenced in a similar manner to the process we described earlier. Usually, in such studies, we are not interested in the identity of each nucleotide, but rather we use the reads to identify each RNA transcript and subsequently to determine the abundance of the molecules, i.e., the level of transcription from every gene. The cost of sequencing has gone dramatically down in the last decade, enabling a revolution in many fields of biological sciences and notably in medicine. Nevertheless, as the equipment and the reagents are still expensive, there is a significant price tag to any large sequencing project. People are already working on more advanced technologies, for example, passing the DNA fragments through a nanotubes in which the identity of each base will be read based on small changes in the electric field, as each nucleotide has slightly different electrochemical composition. This technology will not require chemical reactions and thus will eliminate the need to use expensive reagents. Such novel sequencing methodologies will make sequencing even more affordable and increase the potential applications. In the next segment, we will talk about the computational aspect of the process of aligning millions of reads to the genome.